So let's talk a little bit about each of those groups on a nucleotide. The phosphate group, which again is attached to the 5th carbon, is negatively charged at a neutral pH. And you saw it previously. Our phosphates are usually drawn as such. So here's the oxygen that's attached to our sugar molecule. And you'll see that this phosphate group actually tends to have a -2 charge on it. That's because each of these oxygens here and here are negatively charged. Again, this phosphate is typically attached to the 5' position of the sugar that makes up our nucleotide. All of our nucleic acids, when we start looking at DNA and RNA, are built using a 5' triphosphate version of the nucleotide. What does that mean? Again, it means the phosphate is attached at the 5' position and that there are 3 phosphates. So not just one phosphate group, but 3 phosphates. And we can have numerous versions of this depending on what our nitrogenous base is. This can be called ATP, GTP, TTP, or CTP. Here, the A, G, T, and C just refers to the nitrogenous base, and TP refers to the fact that it is a triphosphate. These triphosphates are useful because 2 of the phosphates that are attached to that whole nucleotide are actually just utilized for building the nucleic acid, for chaining it together. And again, we'll see this when we talk about our nucleic acids in their polymer forms. But those 2 phosphates are utilized just as energy for building, and they actually leave. They form a leaving group and detach, leaving just that one phosphate that was shown previously as the phosphate that is still there on the nucleotide moiety when we build our nucleic acids. But one thing to note is, again, I've been talking a lot about this phosphate on the 5' position. This phosphate can also be attached in certain circumstances to other positions, and that gives it special functionality. So here's an example just to show you some versions of a nucleotide. Here's our normal adenine nucleotide, where we have the nitrogenous base, which is not drawn here, but it's just written as adenine, is attached to the 1' position of our pentose. And you can see on the 5' position, we have our phosphate. Now, you can also have the phosphate attached to the 2' position, the 3' position, or in the case of a cyclic nucleotide, the 2 and 3' position. And this forms this inner cycle right here, which is where we get the cyclic name from. This particular case was actually just given as an example prior. Cyclic AMP, that's cyclic adenine. This is the adenine monophosphate, only has one phosphate. Looks like this and is used to remember as a signaling molecule.